Okay, can somebody confirm that you're hearing me?
Hi, was the sample journal a sample of the notes or the actual report?
Can you repeat that?
Was the sample journal a sample of the notes or the report?
Cancel journal. I'm not sure what you're referring to.
Oh, like the sample journal for the unknown?
Oh, the sample journal. Ah, okay. And it should be on the Canvas website. What about it?
Was it like the notes we were supposed to turn in, or was it the report we were supposed to turn in?
That's for the report you're supposed to turn in. It's an example.
Okay, thank you.
Okay. All right. Are there any other questions? You probably can't see this, so let me show my screen. Boy, my computer is slow today. I'm not sure what the problem is. It's not showing the share screen. Let me try that again. There we go. Hopefully you can see the screen now. Today is March 3rd, and we're supposed to be finishing viruses, viroids, and prions. I'm not sure that we'll finish it, but we'll get close to it. There is no lab today. You should be working on your unknown notes pages and your unknown project. Your unknown project is due Saturday, 11:59 p.m. this week. The unknown notes pages closes on Friday, March 4th at 11:59 p.m. With your unknown notes pages, I did mention last time, make sure you finalize the unknown notes pages so you can get all of the 5 points. Some of you have not put in the results from the last test and given your conclusions and then stated what your unknown bacteria is. Okay, you need to do that to get all of the 5 points in your unknown notes pages. Any question about any of that? Remember, the final is due Thursday, March 10th. It's due at 8 p.m. You take the final online. It's going to be heavy coverage of chapters 7, 13, 14, and lab module 9. So 50% of the questions will come from this area. And that's because you haven't been quizzed on any of this material yet. Chapter 7, hopefully we will cover that next Tuesday. Any questions about the final? It'll be largely multiple choice questions, but there will be a few either fill in the blank or short essay questions. And it's something like 4 to 6 short answer questions, and then the remainder multiple choice. You have 2 hours to work on the final. All right, no questions about that. I'm going to close this down and maybe that'll free up some computer space.
What's quiz 5C on the gradebook?
Can you repeat that? I'm not hearing you too well. Let me turn the volume up here.
Okay, what's quiz 5C on the gradebook?
Oh, that's just the correction to quiz 5. If you notice, quiz 5 was only worth about 1 point or less. And that's because only 4 of the quizzes count towards your grade, and your lowest quiz will be dropped. And so if I had all the points in there for quiz 5, it would overinflate your grade. You would have 50 extra points, and then you would say, oh, I've got an A, and then I would remove the quiz that's not counting your lowest quiz, and that would drop your grade. And instead of doing that, I've got it set up where I will put in quiz 5 in quiz 5C. So the C stands for correction. Okay, so you'll get your points from quiz 5 in quiz 5C, but I haven't dropped the lowest quiz yet. And that will be in the drop quiz column. All right, if there's no further questions, let's go ahead and begin chapter 13. Come on. So we had talked about the virus life cycles, and let me go ahead and open this. Let me turn down my volume now. It's too loud when I'm hearing from the computer. Oh, I don't have that link on this computer. Let me see if I can get it from Canvas. The link didn't work on my other computer, and that link isn't working on this computer because I don't have the file on this computer, and with my headset in, I can't put the thumb drive in. So anyone remember what week we're on? Let's see if I can talk. So we're talking about the lytic life cycle, and it has 5 stages: attachment, penetration, biosynthesis, maturation, and release. And that still hasn't come. I don't know why my computer is so slow today. All right, we're going to come back to this and hopefully it'll work. I need to show you the movie on the lytic life cycle, but let's go through the lytic life cycle. Initially, it begins when the virus attaches to the bacteria, and the virus binds in this region of the virus, meaning the ends of the tail fibers. The virus, after it binds, attachment, it then penetrates. And what it does is the base plate of the virus removes the cell wall in this area. And how it does that, it secretes the enzyme lysozyme, which just degrades peptidoglycan right here. And then the sheath contracts, which works like a needle and syringe, and that injects the nucleic acid of the virus in the head into the cell. And that's known as the stage of penetration. When the nucleic acid gets injected into the host cell, it then will make copies of itself and then be transcribed and make the viral protein. And that'll largely be the viral proteins to the capsid, the capsomeres, which will then, so that's biosynthesis. And then it will undergo maturation, which looks like I've removed that from here, where the viral proteins will come together, forming the virus. The nucleic acid will be stuffed in the head of the virus, and that's known as maturation. The final stage is release, where the virus releases the enzyme lysozyme, which will degrade the cell wall. And, oh, there's maturation there. And when the cell wall is degraded, water can move into the cell, and then the water will burst the cell, meaning pop the cell membrane, and that will release the viruses of the lytic life cycle. There's two terms you need to know: burst time and burst size. Burst time is the time it takes for the virus, once it infects a cell, to the time it takes to lyse the cell. And that's the burst time. The burst size is the number of viruses released. Boy, I can't get this to work on this computer. Let me download the file. The burst size will vary on the species. So the number of viruses coming out of a cell will be something between 50 and 200, but it does depend on what species of virus we're referring to. Any question about the lytic life cycle? Now what, I want to go to slide 19. Let's watch a little movie on the lytic life cycle. Oh, that doesn't look good. Oh, darn. That's going to my computer. That's not supposed to be that way. Ah, fudge. You're not going to get to watch this movie. That link's in error. And that file isn't on this computer, so. Let me see if the next one's also in error. Nope, that's coming from C also. All right. You're going to have to live without my headphone for a moment. And I don't need this anymore. Finally got the movie. Oops, it's not going to play right.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the muphages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA.
All right, let's stop right here and let me talk about the next topic. And like I said, there are two life cycles for bacteria, the lytic life cycle and the lysogenic life cycle. These are two ways that the virus can reproduce. Any questions about any of that? In the lysogenic life cycle, the virus does attach to the cell, and then it injects the nucleic acid into the cell like before. But now one of two things can happen. Immediately, once the nucleic acid gets in the cell, the virus has to decide, does it want to follow the lytic life cycle, or does it want to follow the lysogenic life cycle? If the virus decides to enter the lytic life cycle, it follows the lytic life cycle we just discussed. Any question about any of that? And only lysogenic viruses, lysogenic phages, or bacteriophages, can proceed this way in either the lytic life cycle or the lysogenic life cycle. If the virus decides, once it's infected a cell, to enter the lysogenic life cycle, it does not enter the lytic life cycle. What happens is there's a lysogenic protein that's a repressor protein, and it represses the viral genes of the lytic life cycle. And as long as that repressor protein is active, the lytic life cycle will not occur. The virus will enter the lysogenic life cycle. What happens in the lysogenic life cycle is the DNA of the virus will integrate in the host cell's DNA. And the host cell will remain alive for as long as the virus stays in the lysogenic life cycle. Any question about any of that? So once the lysogenic virus infects a cell, immediately the virus has to decide, am I going to enter the lytic life cycle? In which case it will replicate viruses, lyse the cell and release more viruses. Or it can enter the lysogenic life cycle. And what it does there is the viral DNA integrates into the host cell's DNA. And we call that a prophage, the viral DNA inside the host chromosome. And the only time the virus replicates is when the cell replicates, meaning the DNA of the chromosome will be replicated. That'll make 2 copies of the prophage, and then the cell will divide, and each cell will get 1 chromosome, including the prophage of the virus. And that's the lysogenic life cycle. We'll talk about the rest of the lysogenic life cycle in a little bit. Any question about that? If not, let's watch the movie on the lysogenic life cycle.
Times, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any questions about the movie? If not, let me shut this down. So after the cell divides, and it could go through many cell divisions, every once in a while, the prophage comes out of the host chromosome. And let me go ahead and put in the headphone, because it sounds better. The prophage comes out of the chromosome, the lysogenic life cycle ends, and the lytic life cycle will begin. And then viruses will be replicated, the cell will lyse, and then viruses will be released to infect other cells, bacteria cells. Now, the reason for why the lysogenic life cycle ends and the lytic life cycle begins is not well understood. And they've been studying this now for 50 or 60 years, and they still don't have a good handle on understanding why the lysogenic life cycle ends and the lytic life cycle begins. They do know that one thing that can switch the life cycle from lysogenic to lytic is if you damage the DNA of the host, the virus somehow knows that, and it'll switch from the lysogenic life cycle to the lytic life cycle because it knows the host DNA is damaged, that the cell may die. But other than that, we don't know why the lysogenic life cycle ends and the lytic life cycle begins. All right, any questions about the lysogenic life cycle? I already talked about that. I talked about the precursor protein, talked about that and that. I think I've already talked about that slide. There are some consequences for lysogeny. A lysogenic cell is immune to infection by the same bacteriophage, meaning if this cell has a lysogenic virus in it, it cannot be infected by another lysogenic virus, at least of the same species as the virus that's already in the cell. Okay? Another thing that can happen to this host cell is that it gains new genes, it gains new DNA. And that can revert the phenotype of this cell. This cell, for example, isn't expressing the lysogenic genes of the virus. And the recombinant cell, which has the prophage in it, is expressing the genes of the virus. And that can bring new genes to this cell. And that can change the phenotype of this cell. That is known as phage conversion, where the phenotype of this cell is converted by the bacteriophage. An example of this is the bacteria, Corynebacterium diphtheria, only produces the toxin, which is the toxin of Corynebacterium diphtherium, when the cell has a lysogenic virus in it, because the lysogenic virus encodes the Corynebacterium diphtherium toxin. So yes, depending how sick you will be from this bacterial infection will depend on if this bacteria is infected with the virus or not. Any question about any of that? All right. So enough about bacteriophages. We need to talk about animal viruses. These are viruses that infect animal cells. And we can classify viruses by the kingdom that they infect, meaning we've already talked about bacterial viruses. We call those bacteriophages. Now we're going to talk about animal viruses, the viruses that infect animal cells. And there are viruses that infect the cells of every kingdom. It's just that we're not going to talk about the other viruses other than really animal viruses and bacteriophages, the bacterial viruses. The multiplication of animal viruses follows the basic pattern of viral multiplication, but there are some differences. The virus will go through attachment, will go through penetration. We'll talk about some of the differences, the penetration. The virus will be uncoated. The virus will go through biosynthesis. The virus will go through maturation. And then the virus will be released. There are animal viruses that follow the lytic life cycle. So they go through the same lytic life cycle we've already discussed, where they replicate in the cell, lyse the cell, and then release the viruses, the newly replicated viruses. But there are other life cycles of animal viruses. They do not have the lysogenic life cycle. They have a budding life cycle, which is similar to lysogenic, but different. And then there's the persistent infection, and we'll talk about the other life cycles that animal viruses have. With the replication of animal viruses, the replication does follow the replication of bacterial viruses, but there are several differences. The one difference that I want you to know is that bacterial viruses can only replicate in the cytoplasm, because that's the only area that really a virus can replicate in the prokaryotic cell, because that's all the area of the prokaryotic cell. But animal viruses may replicate in the nucleus, or they may replicate in the cytoplasm. And then there's a difference for which viruses replicate in the nucleus. We'll talk about that in a little bit. But generally speaking, DNA viruses that are animal viruses replicate in the nucleus, and then RNA viruses that are animal cell viruses replicate generally in the cytoplasm. So you should know that difference. There are some other differences too, and I'm not going to talk about them, except that there is a difference of attachment. It's much easier for a virus to attach to an animal cell. And why is that? Anyone have a guess? Nobody has a guess? Bacteriophages have a harder time attaching to the cell and infecting the cell, because bacteriophages have a cell wall they have to get through. It's much easier infecting an animal cell because animal cells have no cell wall. The attachment of the virus attaches to the cell membrane, and in this sense, it's similar. There's ligands on the virus which attach to receptors on the cell membrane. The attachment and penetration of the virus happens a little different, and it can happen by endocytosis, where the membrane folds in, forms a vesicle around the virus, and then that vesicle is brought into the cell. That's endocytosis. We'll talk a little bit more about that. I should mention, if you have an earlier edition of the textbook, it said that viral penetration happened by pinocytosis. That is incorrect. It's endocytosis. The other way that viruses can enter an animal cell is by fusion. If an animal virus has a, sorry, I'm drawing a blank. What the heck is that called? A lipid bilayer outside an envelope. If it has an envelope, the envelope can actually fuse to the cell membrane. And then when that happens, let's show it here. They fuse, and then the cell membrane becomes part of the envelope of the virus, and then will move around the virus, and then that can open up so that the, which I don't have a picture of it, can open up, and this part will then be open, and the virus can just move into the cytoplasm because the cell membrane now becomes the envelope and then goes around like that. And then this opens up, allowing the virus to come into the cell. And that's fusion, okay? Endocytosis is shown here, where the cell membrane invaginates around the virus. And then the cell membrane will pinch off. And so the cell membrane will reform right here. And then that will make a vesicle membrane around the virus. And then the virus can come into the cell as a vesicle. And then the vesicle will be degraded. The virus will be released. The capsid has to be removed. And then the nucleic acid of the virus can get into the cell. All right, the biosynthesis and encoding are similar. Once again, with biosynthesis, you have to replicate the viral nucleic acid, and you have to synthesize the viral proteins. And then there's maturation. That's where the viral proteins come together, form the new virions, and the nucleic acid of the virus will be stuffed into the virus to make the new virions. Any question about any of that? All right. So the lytic life cycle happens very similar to the lytic life cycle of bacteria, viruses that infect bacteria, following the lytic life cycle, meaning the virus will infect the cell, nucleic acid will be made, proteins will be made, new virions will be assembled, and then they'll lyse the cell, and that will release the newly replicated viruses. And there are animal viruses that follow the lytic life cycle. There are also animal viruses that do not follow the lytic life cycle. And the one we're going to mainly talk about is the animal viruses that follow the budding life cycle. The animal viruses that follow the budding life cycle are all enveloped viruses. And non-enveloped animal viruses tend to follow the lytic life cycle, where they will rupture the cell, although there is a persistent infection life cycle that a non-enveloped or more a naked virus can follow. If it's an enveloped virus, it will follow the budding life cycle. What happens is the virus will replicate its nucleic acid and will replicate the viral proteins. Some of the viral proteins will actually be attached to the plasma membrane of the cell. And these might be glycoproteins or lipoproteins, but we're just going to talk about it as viral proteins. And then the virus will replicate generally at low numbers, meaning you don't have lots of copies being made. You only have a few or one copy being made. And then the virus will assemble, and then it will come up to the cell membrane, and a region of the cell membrane that has the viral proteins. And then that virus will push on the, we're talking about it as the cell membrane, although it can be another membrane, but we're going to talk about it as the cell membrane. It'll push on that cell membrane. Pushing on the cell membrane, invaginating it, the virus will come inside of that cell membrane, and then the cell membrane will pinch off. And then we will have really an external vesicle out here making the virus, and then the cell membrane will reform as it pinches off. This will be the cell here, and then this will be the envelope virus. And you see how the virus obtained its envelope. It's literally a part of the membrane from the host cell. Any question about that? So this right here, the envelope, used to be, in this case, the part of the cell membrane. Envelope viruses can follow the budding life cycle, and they can remove or obtain their envelope from the cell membrane, as shown here, from the nuclear membrane, or from the membrane of the endoplasmic reticulum. All right, any question about any of that? And here we're seeing a cell on the electron microscopic image where the virus is following the budding life cycle, and these buds here are viruses that will be shed from the cell. Any questions about the budding life cycle? Can somebody confirm that I have you? Is everything good? Hello?
We can hear.
Okay, thank you. All right. Viruses have different genomes. We've already mentioned that with the bacteriophages. I'm going to re-mention it with the animal viruses. Some animal viruses have double-stranded DNA. Some animal viruses have single-stranded DNA. Some animal viruses have double-stranded RNA for their genome. And some animal viruses have single-stranded RNA genomes. Remember, when we're talking about single-stranded RNA genomes, you have to mention whether that's a positive strand or a negative strand of the RNA. Each virus type needs to have a slightly different mechanism for its multiplication and biosynthesis. And we'll look at that in a little bit. Let's talk about the DNA viruses first. Let's see if I can blow this up. You don't have to memorize the names here, but this slide is showing you the different families of viruses and then the diseases human diseases that they cause. Okay, so herpes viridae, I'm sure you're familiar with some of the herpes virus. That's the herpes simplex virus type 1, type 2, the genital infections, Epstein-Barr virus, chickenpox, which I'm sure all of you are familiar with, Burkitt's lymphoma, cytomegalovirus, roseola virus. We'll talk a little bit about that one. Anyways, there are 7 families of DNA viruses that cause disease in humans. Adenoviridae is the 3rd virus family that causes human colds, meaning it's not the most common. That's the rhinoviridae. And coronaviridae is the 2nd most common. Adenoviridae also causes human colds, so. For DNA viruses, the DNA is generally copied in the nucleus of an animal cell. The reason why DNA is replicated in the nucleus is because in an animal cell, all of the machinery is present for replicating DNA. You have the DNA nucleotides in the nucleosome, am I saying that right? And the DNA polymerases. For DNA viruses, generally speaking, the capsids and the proteins are synthesized in the cytoplasm. And this is done using the host cell enzymes and ribosomes, and the host cell machinery for translating proteins is in the cytoplasm. And so that's why the virus uses the cytoplasm. If they're a DNA animal virus. Any question about any of that? For the DNA animal viruses, there are no exceptions that I know of, that generally they do replicate in the nucleus. Once viral DNA enters the host nucleus, one of 2 things can happen. The viral DNA can be produced, producing new virions, meaning the proteins will be made. And the next slide will then show you the double-stranded DNA will be replicated into viral DNA. Some of that DNA will be transcribed into viral messenger RNA. The viral messenger RNA will be translated into viral proteins, including the capsid proteins of the virus. The capsid proteins will get together with the newly replicated DNA virus to form the progeny virions. And then the cell will release the viruses. Let me go back. So that's one thing that can happen. Boy, this is taking me a while. The second thing the virus can do is the DNA doesn't have to enter the lytic life cycle. The DNA can actually enter, the viral DNA can actually enter the host chromosome and then remain a part of that host chromosome forever, meaning the viral DNA, if it enters the host cell's DNA, it will become permanently a part of the DNA of that host cell. And the DNA will be replicated when the host replicates the DNA, and then the cell goes through cell division. This we do not call a prophage, because phage is a term used for bacterial viruses. We call it a provirus. So it's a very similar term. And like I said, this is a little different in that if you get a provirus in an animal cell, that provirus stays in the host DNA for as long as that host cell is alive. It never is excised. And let's talk about the multiplication of the DNA virus in more detail. The DNA virus infects a cell, attaches. There will be penetration of the virus, and it will come into the cell either by endocytosis or by fusion if the virus has an envelope. Fusion of the cell membrane with the envelope. The viral DNA will then come into the cell. The virus will be encoded. And if the DNA needs to move into the nucleus, which generally a DNA virus always does, it will then move from the cytoplasm into the nucleus. And then that DNA will be copied, making more viral DNA, as well as the viral DNA will be made into viral messenger RNA. The viral messenger RNA will move into the cytoplasm. The viral messenger RNA will be translated into viral protein. The viral proteins will get together with the newly replicated viral DNA to make the new virions, and then the virions will be released from the cell. If it's following the lytic life cycle, the cell will be lysed, and then the virus will be released to infect another cell. But the virus can follow the budding life cycle, where the virus can then bud out of the membrane of the cell, and that could be the endoplasmic reticulum membrane, the nuclear membrane, or the cell membrane. And then the virus can then go and infect another cell. Any question about the multiplication of DNA viruses in animal cells? If not, let's talk about RNA viruses. This slide is showing you the families of RNA viruses that cause human disease. You'll notice there's more RNA families. I believe there's 12. I lost count, 3, 6, 9, 12, 15. Is that right? 15 families. Let's blow that up. Just to mention a little bit about some of them. The family Picornaviridae causes a number of diseases, such as hepatitis A, poliovirus, and then the common cold. There's a division of the family Picornaviridae that we call rhinoviruses, rhino for nose. This is the most common cause of the human cold, a rhinovirus, which is of the family Picornaviridae. The second most common family of viruses that cause the human colds or the coronaviruses, family Coronaviridae. And this is the family that causes COVID-19, and a lot of people call it coronavirus. That's actually the family name. The majority of the viruses do not cause disease, COVID-19. Most of them cause simply the human cold. Like I said, the second most common cause of human colds are coronaviruses. They cause a few other diseases. We'll talk about that briefly. Any other I want to talk about? Oh, we'll talk about retroviruses. This virus is a little different than the other RNA viruses. And so we'll talk about that with about 3 slides. Orthomyxoviridae is probably not a name you're familiar with, but you should be familiar with this family of viruses because this is the family of viruses that cause the flu. All right. I think that's all. You don't need to know the names of the different families or the diseases they're causing, but this is just telling you the different families, the diseases they cause, the strand type. This is single positive RNA virus, single positive, single negative. Here's a double segmented RNA virus.
I was wondering how fast do viruses mutate?
Sorry, can you repeat the question?
How fast do viruses mutate?
It depends on the family. Like for the polio virus, the vaccine was developed in the 1950s. I think one of them was 1954 and the second one was 1958. We're still using those same vaccines that we made in the 1950s, so that the polio virus is not mutating and changing very rapidly. But then again, the flu vaccines and the flu changes rapidly, so that after you're given a flu vaccine, after 3 years, you're no longer getting protection from that vaccine because the virus changes so rapidly. So it really depends on the family of virus. Some of them do not change rapidly. Others of them do change rapidly. And with the coronaviridae, they change fairly rapidly. Maybe not as rapidly as the flu virus, but it does change rapidly. And so my guess is we'll have to get a COVID-19 vaccine shot about every year, maybe even every 6 months, but probably every year, and certainly every other year. Not stop the share. Yeah, no. End the show. That's what I want to do. All right, here we're looking at some RNA containing viruses. You can note that they're different shapes and sizes. The multiplication of RNA viruses is somewhat similar as DNA viruses. There are some differences because it's RNA as their genome instead of DNA. Generally speaking, RNA viruses replicate in the cytoplasm. There is one exception, and that is the retroviruses, an RNA family of viruses that do not replicate in the cytoplasm. They replicate in the nucleus. Any question about that? Remember when we're talking about RNA viruses, if it's a single strand of RNA, you have to state whether it's a positive single strand RNA or a negative strand, single strand RNA. When we're talking about DNA viruses, it's fairly easy to replicate 2 strands of DNA. One strand is just used if you have a double-stranded virus. One strand is just used as a template to make the other strand. For RNA viruses, that's not the case. A positive strand, single strand RNA virus will replicate itself by using the positive strand RNA to make negative strand RNA. And then the antisense strand, the negative strand RNA, is copied to make more of the positive strand RNA. So to copy positive strand RNA, you do it in 2 steps. First, you make the negative strand, and then you use the negative strand to make the positive strand. The positive strand RNA genome can be used by the ribosomes. It can read it as messenger RNA, and it can be translated into proteins. For the negative strand RNA virus, you make the positive strand RNA virus and then use the positive strand to make more of your viral genome, meaning the negative strand RNA. The negative strand RNA cannot be read by the ribosome, and so you cannot make protein from the negative strand. To make protein, you make the positive strand RNA, and then it can be used as messenger RNA of the virus and will be read by the ribosomes and translated into the viral protein. Any question about any of that? So negative strand RNA virus, you use the negative strand to make the template, the positive strand RNA, and then you use the positive strand to make more of the negative strand genomic RNA of the virus. The positive strand will be used as messenger RNA for the virus, translated into proteins, the capsid proteins to get together with the replicated viral genome, the negative strand RNA, to make the progeny virions. If you have positive strand RNA virus, to replicate it, you make the negative strand corollary RNA, and then use this as a template to make the positive strand genome of the virus. The positive RNA can also be used as messenger RNA for the virus to translate into the viral proteins. Then the capsid proteins will get together with the replicated positive strand RNA to make the new progeny virions. Let's talk about the replication of a positive single strand RNA virus. When this cell infects a cell, it will bind from attachment molecules on the outermost portion of the virus to the receptors on the host cell. And that binding is specific. And then the virus will be brought into the cell, either by fusion, if it's a envelope virus, or by endocytosis. Come on, where's my mouse? And then that will be brought into the cell by a vesicle if it's by endocytosis. The virus is RNA virus, so it generally will stay in the cytoplasm, but there is one family that it differs, and that's the family of retrovirus, and we'll talk about that virus family in a little bit. The positive strand RNA virus will make the negative strand RNA, and then the negative strand will be used to make more of the viral genome, the positive strand RNA virus. The positive strand can be used as messenger RNA to make and translate viral proteins. The viral proteins will get together with the newly replicated viral genome to make viruses, the new progeny virions. And then these will be shed from the cell either by lysis or the budding life cycle or some other life cycle we'll talk about in a little bit to infect a new cell. Any question about the replication of a positive single strand RNA virus? If not, let's talk about the multiplication of a negative strand RNA, single-strand RNA virus. We'll have our virus attached to the cell, be brought inside the cell. The viral RNA will be replicated into the positive strand of the virus, and that positive strand will be used as a template to make more of the genome, which is the negative strand RNA. The positive strand will be used as messenger RNA, viral messenger RNA, to make viral proteins. The viral proteins, at least the capsid proteins, will get together to form the virus, and then the new negative strand RNA will be stuffed in the virus to make the new virions, which will then be shed from the cell. Any question about the multiplication of negative strand, single strand RNA virus? If not, let's talk about the exception, and that is, generally speaking, the RNA of an RNA family will stay in the cytoplasm. It does not move into the nucleus. And that's because RNA has all the machinery of the host cell it needs in the cytoplasm to replicate. But there is one exception where there's an RNA virus family that actually multiplies in the nucleus. It does not multiply in the cytoplasm. Let me open up Microsoft Word. Probably it would be better if I did Paint, but I don't have it open. So this exception is the family of retroviruses. Not spelling well today. Why is that so small? That's better. Retroviruses replicate in the nucleus. Any questions about that? So this is the exception, the one RNA family that replicates in the nucleus. Retroviruses are important because they include the HIV viruses, HIV-1 and HIV-2, which cause the human disease AIDS. And another retrovirus you may have heard of is feline leukemia virus, which causes leukemia in cats, leading to a disease similar to AIDS. It's similar in the sense that the cat's immune system shot to hell, but that's because of the leukemia and feline leukemia virus. And anyways, that's another retrovirus. Retroviruses are unique in that they have the gene for the enzyme reverse transcriptase. This is a protein that there's actually one copy of it in the virus shown right here, the little blue thing. And the reverse transcriptase can take RNA and make DNA from a RNA template. Retroviruses comes from the first two letters of reverse and transcriptase. And so that's why the name retrovirus came about, because it has the enzyme reverse transcriptase in the genome of the virus. Let's see. Let's go here. Move that down here. So attachment, penetration, and coating of the retroviruses is the same as other animal viruses, but biosynthesis happens in the nucleus. It's a positive strand, single strand RNA virus. And if you look here, you'll note that there are two strands that both of them are positive. So we're just going to call it a positive strand RNA virus. And then the reverse transcriptase takes the RNA to make DNA. And the step here is RNA, let's go viral RNA in the presence of reverse transcriptase. Why is that so small again? And I'm not able to get that to go. So RT stands for reverse transcriptase. So RNA in the presence of reverse transcriptase makes single strand DNA. And then single strand DNA in the presence of reverse transcriptase again can be made into double strand DNA, which then will integrate into the host chromosome and become a permanent part a part of the cell. So when the retroviral DNA integrates into the host chromosome, it permanently becomes a part of that cell's DNA. That viral DNA will never leave the cell's DNA. And for as long as that cell is alive, it will have retroviral DNA in the cell's genome. Any question about any of that? Although the so let's go viral DNA plus host DNA can then be transcribed and can make viral, let's go little v, viral RNA, which can be used as messenger RNA to make viral proteins. And then this viral RNA is positive strand, and it can get together with the viral proteins to make new viruses. Which in this family then can bud out of the cell. Finishing the replication cycle of the virus. Okay, so the viral RNA with the reverse transcriptase. First makes single-strand DNA. The reverse transcriptase uses the single-strand DNA to make double-stranded DNA. The double-strand DNA can then be copied to make positive strand RNA, which is actually the genome of the retrovirus. And then this can be read by the ribosome, translated into capsid proteins, which can then get together with the positive strand RNA genome of the virus to make the new progeny virions, which will then bud out of the host cell. Retroviruses are a budding life cycle. They follow the budding life cycle. So let's talk about that in more detail. Let me see if I can blow this up. So the retroviruses attaches to the host cell, and then that virus is brought into the host cell. The viral positive strand RNA will be made first into single strand DNA, and then the single strand DNA will be made into double strand viral DNA by reverse transcriptase. That viral DNA will then move into the nucleus and form a provirus, integrating into the chromosome of the DNA of this cell. And that integration more or less happens at random. The retroviral DNA integrates at random in different places and different chromosomes of the cell. And where it goes will just depend on what cell it is and where the virus happens to integrate. The viral DNA will remain a permanent copy of the cell, and it can be transcribed into viral positive strand RNA, which will move into the cytoplasm to make viral proteins. The viral proteins and then get together in maturation and form new virions, stuffing in the viral genome, the positive strand RNA of the virus. And then this virus will come up to a piece of the membrane where there are viral proteins in it, and then start budding out of the membrane. And it depends on what virus it is. Most retroviruses bud out of the cell membrane. And that's how a retrovirus obtains its membrane, by ripping off a piece of the membrane of a cell. Any question about viral multiplication of retroviruses? All right, no questions. There was something I wanted to say about retroviruses. They are the exception, an RNA virus that replicates in the nucleus. They're an exception and replicate in the nucleus because of reverse transcriptase. And they have an unusual intermediate, a DNA mediate intermediate. And that's why they replicate in the nucleus. All right, any questions about retroviruses and RNA viruses? If not, let's talk about the next family of RNA viruses I want to talk about. Coronaviruses, family Coronaviridae. They are single strand, positive strand RNA viruses. Oops, way too big. Come on. I lost control of the mouse. About there's where I want. They are envelope viruses. They have been causing the human colds for a long time. Like I said, they're the second most common cause of the human colds. So most coronaviruses simply cause the human cold. There are some exceptions. Coronaviruses also cause SARS, MERS, and COVID-19. All of these viruses can cause death. SARS stands for severe acute respiratory syndrome. MERS, Middle East respiratory syndrome. MERS has the highest mortality rate, with the mortality rate being around 36% of patients who are infected die. But the good thing about MERS is when a patient gets MERS, it does not spread from human to human. Most people who come down with MERS get it from an infected camel. Okay. SARS has a mortality rate of around 10%. It can spread from people to people. And fortunately, it actually arose in China twice. Fortunately, people were able to get control of it, and it did not spread to the general population. Okay. SARS is actually closely related to COVID-19, and this data is out of date now. The mortality rate of COVID-19 in the past used to be 0.02% to 3%. It may be less now because Omicron variant is less lethal than the previous variants of COVID-19. The danger of COVID-19 is it spreads very rapidly, especially Omicron, the most common, the prevalent strain of COVID-19 in the United States. And it does spread more easily than the flu. It used to be if one person got infected, they tended to infect between 1 and 2.5 other people. With Omicron, this number is probably higher. Okay. And the reason why I'm not giving you the numbers for Omicron is because we haven't studied Omicron long enough to have numbers yet for what the exact mortality rate is and what the infection rate is. But that they should be getting those numbers pretty soon. Coronavirus, COVID-19 actually has a scientific name, and it's called severe acute respiratory syndrome coronavirus 2, or SARS-CoV-2. And this name is so similar to SARS that even scientists tend not to use it. They tend to use COVID-19. And if you don't know, COVID-19 simply means coronavirus ID, COVID, meaning ID, identified in 2019. That's why COVID-19 got its name, COVID-19. It is a virus closely related to bat coronavirus and likely originated from a recombination of 2 bat coronaviruses and a pangolin coronavirus. The pangolin coronavirus only encodes about 10% of the genome of the virus, coding the spikes on the coronavirus. And they know this from sequencing the virus, that it looks like it recently recombined from actually 3 previous coronaviruses. Probably not all at once. It probably combined once and then combined again with the 3rd. The pangolin is a spiny anteater. So if you want to call it anteater, you can. And the anteater was probably an intermediate, meaning that the coronavirus probably spread from bats to anteaters and then spread to humans, probably from anteaters. And it may have been just one anteater that had the COVID-19 virus. And then probably that anteater was brought into the Chinese market because they ate a whole bunch of different animals. And Wuhan in China had the market. And then the COVID-19 spread from the anteater to a human. And that's why the first identification of COVID-19 happened in Wuhan, China, at least as far as we know. Anyways, that's the likely story for what happened and how COVID-19 came about. In the past, COVID-19 did not exist. And fairly recent, we don't know when, it was a recombination of 3 coronaviruses. Just to show you this virus here, it's actually the virus I use on my webpage for, what is that called? Canvas, is that what it's called? Canvas. And I grabbed this many years before COVID-19 was ever discovered. This is actually a picture of a coronavirus, and I thought it was a cool picture, and so I grabbed it. And now it's a picture of COVID-19 because it's one virus species in the coronaviruses of family. Any question about any of that? All right, let's end up the COVID-19. COVID-19 is primarily spread droplets from a cough or a sneeze within a range of 6 feet, but you can get it even from the person breathing or singing or talking. Talking and singing are much more common than breathing, but you can shed it by breathing. It's likely most often breathed in, but you can pick it up from an object and then pick it up on your hands and then inoculate a membrane, a mucous membrane in your face. It's just that COVID-19 seems to spread directly from a person to another person from droplets. It can survive for 3 days on plastic or metal surfaces, but it can survive for 1 day on paper or cardboard. Okay, any question about that? The symptoms of COVID-19 are mostly flu-like, though runny nose and sneezing are less common, and shortness of breath and deaths are much more common than the flu. The mortality increases with age, ill health of the patient. Mortality is more common in men and Blacks and Hispanics. And this is not understand why Blacks and Hispanics have a higher death rate than whites and Asians, but it is seen in both the United States and in Mexico and in Canada can confirm this. And then all countries can confirm the deaths are slightly more common in men than in women. The incubation period, you're infected with the virus 2 to 14 days before you have symptoms. Some people remain asymptomatic. Among young adults and children, it's estimated that young adults and children, about 50% of the patients are asymptomatic. And that's how it came into this country, by the way. An asymptomatic carrier brought it into this country. The estimated death rate is between 0.2% and 3%. You can help prevent it by correctly washing your hand, keeping your distance from people, especially if they're coughing, not touching one's face, and by wearing a mask. It's best to stay home if you're sick so you don't spread the disease. All right, any question about any of that? If not, I'm going to end it here and we'll continue this next time. Okay?
I have a question about the unknown project.
Okay.
Yeah, I have one more test that needs to be ran for the unknown project. Is there any way you could do this today or tomorrow?
How long does the test incubate?
I'm not sure. It's the...
Okay, put in your request and I will send results for Friday for sure.
Okay.
Okay. Somebody requested tests yesterday, and I think I'm going to send requests after the lab time today.
Okay, thank you.
Okay, all right. All right, see you guys.